Good morning, everyone. Our office has been involved with invasive species monitoring since the mid-1990s. And recently, the U.S. Fish and Wildlife Service expanded efforts to detect invasives across the Great Lakes. And today, I'll just be providing an overview of our monitoring efforts in Lake Huron. It is well known that over 180 non-native species have become established in the Great Lakes, and a number of other species shown here are at risk to invade the area. There have been some species-specific early detection and monitoring efforts, but a real need exists for coordinated early detection and monitoring across the Great Lakes. Early detection may allow rapid response to curtail or prevent the spread of newly discovered non-native species and their impacts. With funding provided by the Great Lakes Restoration Initiative, the U.S. Fish and Wildlife Service Fisheries Offices initiated a comprehensive program for early detection and monitoring for bighead and silver carp and for non-native species new to the Great Lakes. The program involves sampling for bighead and silver carp environmental DNA, larval, juvenile, and adult non-native fishes, and non-native benthos. Each of the Great Lakes Fish and Wildlife Conservation Offices are implementing this program within their respective jurisdictions. I'll be touching on each of these elements conducted by the Alpena Fish and Wildlife Conservation Office. One element of our aquatic invasive species program is monitoring for the presence of Asian carp environmental DNA. Environmental DNA would include any genetic material that may be present in the environment, in this case the water. Water samples are collected in the spring at tributary locations chosen in cooperation with state management agencies. Samples are then analyzed for the presence of bighead and silver carp at the service's Whitney's Genetics Lab in La Crosse, Wisconsin. Our 2014 efforts are shown here. There were no positive detections. Another element of our program is the early detection of non-native fishes and benthos. We have developed lake-wide implementation plans to guide efforts to detect these species within our jurisdiction of Lake Huron and also Lake Erie in cooperation with the Lower Great Lakes Fish and Wildlife Conservation Office, where we share jurisdiction. We have identified 33 species that are at risk to be introduced into the Great Lakes, including 6 amphipods, 3 bivalves, and 24 fish. However, our implementation plan and sampling is not targeted on any one species or is non-species specific. An important part of the implementation planning process is a vector risk analysis that examines pathways for entry of new species into each lake. Sources within each of 8 invasion categories shown here were examined, including data such as tonnage of ballast water discharged at ports, number of bait shops per capita, among others. Vector categories were then weighted based on historical or past invasion pathways for established invasions in the watershed and ranked by port or tributary to identify priority sampling areas. Source data is constantly updated through time, and vector rankings and priorities may shift. This map shows our vector-based priority sampling locations for Lake Huron and also Lake Erie. Due to limitations on time, staff, and resources, we identified 4 locations for sampling, shown outlined in red. These areas include the lower St. Mary's River below the compensating works. Also, other locations in Lake Erie, including the Detroit River, Maumee Bay, and Sandusky Bay. Our sampling approach for juvenile and adult fishes follows research by Trebitz et al., who examined maximizing gear efficiencies to detect rare and non-native species in the St. Louis River of Lake Superior. The Ashland Fish and Wildlife Conservation Office in Wisconsin implemented this approach early on, and we are following for Lake Huron and Lake Erie. The premise is that newly introduced species will likely be low in abundance or rare within a system. The strategy is to detect the presence of all known species or an acceptable percent of known species using a variety of sampling gears. Gear effort will be maximized towards gears that detect a greater diversity of species and those that detect the highest number of unique or rare species. Initially, phase 1, we are using a stratified random design with equal gear allocation between depth strata in an effort to detect the presence of known species. Gear performance will then be analyzed and effort will be allocated in phase 2 to gear mixtures that maximize rare species detection. The lower left image shows species accumulation with an increase in samples. Common species are detected early and more unique species are detected with greater effort. The lower right image shows the adaptive process of evaluating gear performance and recommending gear mixtures to maximize rare species detection. This shows our sampling locations in the St. Mary's River in 2013 and 2014. We used paired fyke nets, nighttime electrofishing, and bottom trawling gears in equal allocation of effort. We suspect it may take 3, 4, possibly 5 years of sampling in order to collect an adequate number of samples that will approach a 95% detection rate for rare species. It's sort of yet to be determined. This sampling was conducted in partnership with the Ontario Ministry of Natural Resources Upper Great Lakes Management Unit, who conducted some of the sampling in Canadian waters. This shows our sampling gear performance through 2014. Fyke nets and electrofishing basically collected the largest number of species, and actually most of the species that were present. I think the trawling actually did collect 1 unique species, but predominantly fyke netting and electrofishing have been the better performers. No new non-native species were detected. We've captured 42 species out of 84 samples to date. I also wanted to mention that this sampling effort is ongoing in Lake Erie at our other priority locations. This shows preliminary species accumulation curves for our sampling areas. We are also conducting sampling for larval fish and benthos in priority areas on Lake Erie. These efforts may be expanded into Lake Huron at some point following vector-based priorities. Our office is merging existing species-specific sampling efforts into our new comprehensive early detection and monitoring program. We continue to conduct targeted efforts to detect Eurasian ruff in Lake Huron as part of a coordinated effort that's being conducted in conjunction with the Lower Great Lakes Fish and Wildlife Conservation Office, who is sampling on Lake Erie and Lake Ontario, and also in cooperation with many partners across the basin that are providing information on their incidental catch for ruff. Our sampling targets harbors, ports, and rivers using bottom trawling gear. This program was initiated within Lake Huron in 1995. Recent sightings of ruff since 2008 in northwestern Lake Huron have resulted in an expansion of our efforts in these areas to include trap netting and electrofishing as a means to aid in learning more about ruff in these areas. Despite occasional sightings, we have not detected ruff in our sampling gear in some of these other areas where they've been newly sighted. The last time that we did capture ruff within Lake Huron was from the Thunder Bay River, where they were a part of the fish community from 1995 through 2003. We continue to sample that area, of course, looking for ruff, but we have not detected them. There was some, University of Notre Dame and the Nature Conservancy conducted an EDNA study to look for Eurasian ruff DNA across the Great Lakes, and they did sample within our area of coverage of the St. Mary's River and also northwestern Lake Huron. And there were some detections in these areas, so part of our follow-up is to learn whether this eDNA that was discovered in these areas is from actual fish. So we have been sampling in those areas, and they include the Sheboygan, the ones that are listed here, the Trout River in Rogers City, and also the Thunder Bay River. There's also a river just south in Thunder Bay, the Devil's River. They did have a finding there. So I think the findings were the Devil's River, Sheboygan, and the Trout. They did not find DNA in the Thunder Bay River. We would just like to acknowledge